Hi and welcome to my fourth video in the series about microorganisms and health, and today we're going to be looking at antibodies and memory cells. So we're going to look today at how antibodies, first of all, how they work against specific antigens. And the first thing to notice on this diagram here is the green parts represent a foreign pathogen, and these surface sections kind of in yellow, in a kind of yellowy colour, they represent the antigens on the surface of the pathogen. Now the antibodies are represented by these structures here, and you'll notice that the antibodies have a complementary shape to the antigens, and therefore they will fit. So certain B cells will produce different types of antibodies, and therefore only a specific type of antibody will fit a specific pathogen. And so that's what takes the time in order to find the correct B cell, and therefore to produce the correct antibody to fight a specific infection. Now, antibodies can work by clumping together big groups of pathogens, whereas with a single phagocyte they can only encounter possibly one pathogen at a time. By clumping all of these pathogens together, it makes it very easy to bring in a very large phagocyte, or what's sometimes known as a natural killer cell, to come in and to basically wipe out the entire population very quickly. And that's why we produce the antibodies, because they're very effective at clumping together loads of pathogens. They also work by deactivating and stopping the pathogen from reproducing anymore, and hence stopping the growth of the infection. One final note on antibodies: there are actually 5 specific categories: A, E, I, G and M. But with any given infection, because their antigens will be subtly different, there are thousands upon thousands of different types of antibodies that your body can produce, all with very specific shapes. You can think of it much like enzymes having a specific lock to go with a specific key. Now, the time taken for you to reach the right B cell that produces the specific antibody to fight a specific pathogen will take some time. And that's the time in which you can actually begin to feel ill, because what can happen all the time that you're not finding the correct antibody that can deactivate or clump together large numbers of these pathogens, then they can basically make huge numbers of copies of themselves, and hence bacteria can make you feel ill by producing toxins. Viruses can actually cause direct damage to your cells when they infect them. But what happens once you've found the correct B cell, as in here? It then differentiates into 2 different types of cells. The first of those cells is called a plasma cell, and the plasma cell's job is just to pump out millions upon millions of antibodies into your bloodstream, and therefore you can clear the infection up quickly. The second type of cell is what's known as a memory cell, and a memory cell is there for when you get a secondary infection with the same pathogen. Those memory cells will stay in your system, and when you encounter the exact same pathogen again, those memory cells will turn into plasma cells very quickly, and then those will produce huge amounts of antibodies. And so what will happen is you'll combat the infection much quicker. This graph shows us what an immune response will look like. Along the bottom you've got the number of days post-infection, and then up the y-axis you've got the number of antibodies. And so you can first see here, what happened here first is the first encounter with a pathogen. So maybe at roughly day 2 or 3, it's the first time you've been infected with the pathogen. And then you'll see it takes a reasonably long amount of time, so several days, so up to 5 to 6 days before the antibody numbers begin to increase. And this is what I was talking about before, where you have to find the correct antibody, and that taking some time. Now, after the first encounter, you'll produce memory cells, and so on the next occasion on which you've been infected with a pathogen, so in this case here, you'll then see that because the memory cells are within your system and are there very quickly, they can differentiate very quickly into plasma cells, and you get a much bigger immune response, so you produce more antibodies, and it's much quicker. The period of time that it takes from first infection to produce the antibodies in any number is called the latent period. So on a secondary response, the latent period is much shorter, and you produce a huge amount, or a much bigger amount of antibodies than you would do with a primary response. And hence why, when you come across the same pathogen again, you don't feel ill. So in summary, antibodies, they bind to the surface of antigens on a pathogen, and they have a complementary shape. So a specific antigen on a specific pathogen will only have a specific antibody that fits it, and hence why the shape is referred to as complementary. How they work is they clump together big numbers of pathogens, and then now you can bring in something like a phagocyte or a natural killer cell, and they can come in and destroy large numbers of the pathogens. They can also work by deactivating them and stopping them reproducing. So they're very, very effective at producing an immune response. The B cells can differentiate into two types, so plasma cells, and those are the ones that produce the antibodies, and memory cells, ones that stay in your system ready for a secondary response. So if you were to get infected with exactly the same pathogen again, you have your memory cells there for a quick secondary response. And within a secondary response, there's a larger number of antibodies produced and a shorter latent period.